So, yeah, Hannah pretty much said it. It's cool to be here talking to you guys, especially from the research side of things. I titled this presentation "Growing Pains" because that's really going to be kind of the theme of this is how we've grown both as a research group, but also within the LabKey software and what it's enabled us to do, and some of the new capacity that we're using it in since last year. And we're going to talk about scale. So I think something that our lab group does that's not necessarily unique, but definitely different than a lot of other research is the scale at which we collect samples and run assays. And it comes with really unique challenges, both collecting that many biospecimens, but also storing data from them. We're going to talk about challenges that brought up and how LabKey has helped us solve them. So who are we? Hannah said it, the AIEERP. It's kind of a mouthful, but Dr. Bowman, who presented last year, is our PI. We also have Dr. Nolting, who's our co-investigator. And then these are our full-time staff members who really make the ship sail. Heather and Deborah have been super key on the laboratory side of getting things set up. And Avery is a new research assistant with us who, without in the last talk, there was a question about buy-in, right? And so without your laboratory staff really buying in and getting into how to use this, it doesn't fly when Andy and I are the only two people answering LabKey questions. And so it doesn't happen without the whole team. I'm going to talk a little bit about what we do since it's a little bit unique. So we focus on zoonotic diseases, which are diseases that transmit from animals to people, right? And so we do this kind of surveillance at these interfaces where transmission can occur between animals and people, and then use those data to produce and inform preventive strategies that allow us to protect animal and public health. And a big part of what allows us to do that is active disease surveillance. So what's kind of involved in this is identifying animal reservoirs for diseases, particularly those that present a high risk. If you're thinking about it from a public health perspective, you want to look at animal reservoirs that can either transmit directly to people or host a viral diversity that can eventually spill over to people, or that just have a lot of the disease in general. We're going to identify those animals and then go out and prospectively collect samples. So we're not necessarily getting animals into a veterinary clinic, even though we're at the College of Vet Med. We're going out to an otherwise unstudied or healthy animal population and collecting samples from them ahead of time before a disease outbreak. We're going to test them for whichever pathogen we're looking for. And then once we get those data, we're going to analyze the epi of that disease that allow us to do those strategies that can help us improve animal and public health. And so to talk about how we do surveillance, I want to give you guys a picture of the natural structure of the data that we collect, because it's a little bit challenging. So previously we focused almost exclusively on influenza. I'll call it IAV. And our first study from our emeritus PI was on avian influenza. So wild waterfowl are the primary reservoir for flu. We've been doing this since 1986. So you can imagine how much things have changed since 1986. These are primarily going to be hunter harvested birds, but it includes some other smaller projects along the way, like birds in zoos or live trapped or poultry and other host groups like that. We have over 70,000 samples collected since 1986. And then since 2009, if you remember the 2009 swine flu pandemic, we also started looking in pigs. In the United States, as it turns out, swine at county fairs and exhibitions are the primary zoonotic risk for flu in the United States. And so swine are this unique host that allow for reassortment and pandemic risk. And so we've been doing this since 2009, mostly from exhibition pigs. And we have over 60,000 samples to date. So much shorter time period, but as you can see, a lot more of higher intensity surveillance. We had a couple of projects that really involved intense sample collection. But so what this looks like is a lot of waiting around, right? So this is avian surveillance, and what it entails is taking a tote with gloves and vials and swabs and sitting out at boat ramps where we know people launch boats to go duck hunting. And we sit in the parking lot and we wait for hunters to come in. And then we ask if we can swab their ducks, mostly cloacal swabs like this. And we record metadata. And so The interesting part about this is how it's clustered. You can imagine in disease transmission, they cluster spatially, but we also have these distinct sessions in which we're going out, right? So like on any given day, we're collecting metadata about the wind speed and other bird species we see around. For some projects, we collect water salinity and pH, and we measure turbidity. And so for any one of these individual days, we have all of these metadata that are associated with those samples that get brought in on that day. And then we might be back out the following day, which has its own session with its own unique metadata, in addition to individual level data associated with the ducks, such as age, sex, and species. So you can probably picture the structure of clustering, right? We have multiple levels of metadata that we need to keep track of that can pose a challenge, especially over time. And then pigs at county fairs, it's as silly as you might have imagined. No, we don't actually get to attend the fair all that much, but every now and then I cannot resist a good bucket of donuts photo. So these are our pigs that are brought to these fairs to be shown either as a part of a 4-H project or, depending on the show, sometimes just for the competition of it all. And so we roll in and we collect either nasal swabs or snout wipes from these. Same sort of data structure though, right? Not only do you have clustering within states or counties, but we collect, let's say, 20 samples from within one show. Those are obviously not independent of one another because you have pigs living under the same barn for a week at that county fair, right? That fair has individual data associated with it, such as organizational practices and how long that show is. And then pigs have breeds, individual clinical signs, age, and things like that. And so where did this leave us? Well, up until somewhat recently, just before the pandemic, it left us with two pretty much just completely distinct databases. It's easy enough, two simple sampling projects. We go out, we wait for animals to come to us, and then we collect our samples. But the data structure is not super well represented with just a simple data set, right? We can record all of the cluster level data and all of the animal level data, but it really only gives us the ability to view that as a simple table, right? With either in the long format with duplicated metadata, or the wide format that makes it more difficult to look at individual level predictors. It was pretty hard to use, especially I joined our lab program in 2015 as an undergraduate student. I was 17 at the time, and learning how to use this database was really difficult. It also only provided us local access, as it was managed by our local IT department at the College of Vet Med, which made it difficult to access data when we were not actively in the office without VPNing into a workstation on site, which made it difficult to both answer real-time questions, or if you're in the field and you want to double check things, or even if you want to enter data along as you go, it makes it really hard to do that. Another huge challenge I sort of hinted at is the longevity of it. You can imagine how much data types and laboratory assays have changed throughout the surveillance period going back to 1986. You may have heard of H1N1 or H3N2. Those are antigenic subtypes of influenza virus. Until somewhat recently, that was done with actual antigenic characterization, but recently those get classified using a genome sequence of the virus. So it's a genetic classification of the antigenic segments, right? And so we have a whole bunch of samples that are antigenically classified and a whole bunch that are genetically classified, but the H1N1 looks the same, methods just change. And so it's really complicated to keep track of what PCR assay were we exactly using in 2001. And neither of these databases are really well suited to accommodate new research goals, right? So new species, new projects that have different metadata that you want to record and things like that, really just no room for growth. And as we were hitting this growing pains session, really just before the pandemic, there were a lot of things that made it clear to us that it was time for us to move on. This turnover of personnel is really big for us in academia. A lot of students come and go and are really only around for a couple of years at a time. We also happen to be losing two of our long-term full-time research staff members who had previously had a lot of experience with the old databases that were now going. And so we were sort of vacating some of the old stubborn, well, this is how we used to do it groups, and getting in a lot of new people that made it a little bit easier and encouraging to switch to something that might work better in the long term. Our bird surveillance is getting scaled down. Our swine surveillance is going to continue. We're starting a longitudinal human cohort, which comes with a whole scope of its own problems, another layer of clustering now that we're adding individual human subjects that are being sampled over time. They're also being sampled at swine shows, so you have that additional data level to account for. plus all of the different sample types that are going to come from them that are unique compared to our birds and pigs. In 2021, we discovered SARS-CoV-2 in white-tailed deer, which was a bit of a whirlwind, but so it has sort of kicked off this entire field for our lab group, which is swabbing white-tailed deer to see if they have SARS coronavirus 2. So we have a new species, we have a new pathogen of interest, completely different assays, especially as the PCR and sequencing assays for SARS-CoV-2 kind of developed in real time during this pandemic. And then what about SARS-CoV-2 and mink? We've heard about the mink problem. We've heard that there's money to address it, and we are trying our best to do so, but it's a little bit tricky and it poses its own challenges. And so when we went to look for a new software, we want a lot of sample and source types. Right across our study, we collect really different metadata. What we collect for bird samples is really different than what we collect for swine samples. And so we need to have flexibility to be able to address those. And then a lot of assays. It varies by project and it varies over time. Sometimes we get free assays from the CDC to test the project that they fund, but we use a different assay for swine in a slightly related project. And we also wanted it to be accessible, right? So cloud-based allows better access. Wanted it to be easy for our new students, especially our undergrads who might not have a lot of database experience to learn. Compliant with our institutional security requirements, which kind of seems like a gimme, but I don't know if anybody here really loves working with their university IT department. I know I certainly don't. They were managing our old databases and it took forever for them to fix anything. And so getting something that we could really manage ourselves, but was still compliant with those requirements was important. And these question marks are the big thing. So I listed a couple of pretty basic needs, but I think the best part about working with LabKey, at least from my perspective to date, is how things come up that we didn't necessarily know we needed or could make use of that we have found to be really useful. So we work very closely with Hannah as well, just like Sydney mentioned. And we just never really feel like we're being hung out to dry. If we have issues, Hannah both addresses them extremely rapidly, but also every time the new patch updates come out and we see the list of new changes, it's like, oh yeah, that actually seems really useful. And I think that's one of the real pros to having a software like this, where Hannah works with so many different researchers and has that insight to issues or obstacles that researchers in a lot of different fields sort of come into with their data, and then sort of fielding all of those notes and complaints or comments and being able to put them towards development really ends up with something that is greater than the sum of its parts, because it's a lot of things that you didn't necessarily look for to begin with. And so here's where we're at right now. You can see we have a lot of sample types. Some of these are species specific, and part of the challenge for that was figuring out how exactly the assay and the cluster level metadata walk through the table within the LabKey. And it was really useful in helping us understand what data can be walked to different levels. You can see that our human cohort really has just started. There's not a lot of samples compared to our ancestral avian data. And then nasal wipes and nasal swabs really are both of the sample types that are focused on our swine. And then, of course, the mink project has really taken off. We've collected a ton of swabs just this summer for the SARS-CoV-2 mink projects. And then these are some of the overflow from previous projects: environmental wipes, air samples, aerosol samples, lymph nodes, and things like that. And then since the last time, probably, we've really focused on expanding and integrating our assay data. Our project critical assays are PCR. So this is the first thing that gets set up. You can see we have our flu screen, all of the different crazy SARS-CoV-2 panels, and then some microneutralization assays here as we get through. We're kind of balancing both being able to get our data in as it comes so that we don't lose it or have to temporarily store in Excel files, as well as working on the backlog of all of those years of data that we want to transfer over. Working with Hannah to make our layouts so that our data reporting is easier through both our funders and our collaboration networks, and then new assays on the horizon. And like I mentioned, the old ones that still need to get put in. Something we didn't really have on our list when we were looking, but has turned out to be really useful is the storage management. We're not really making full use of this yet. You can see we've only got about half of one freezer logged, but we're up to 13 freezers and 4 liquid nitrogen tanks full of biospecimens and different viruses. So not just original samples, but also viral isolates that are from those parents. And so this is our freezer room, and it doesn't really do it justice of having that one freezer from 1992 that just broke down and we had to find spots for all of those boxes. We have outdated paper box maps that are from old freezers that aren't here anymore, or boxes that have disappeared or been thrown out or sent to a collaborator. And so reconciling these and really inventorying what we have and getting it into storage is a Herculean task. But we're working on it very slowly. And although my time is limited here, as I look and turn my sights towards graduation, I know that this is just going to be really, really great for the lab program in the future. And then again, looking to the future, these are two more things that were happy surprises, right? We have new laboratory management and frequent turnover. And so we need to simplify the things that we do on a regular basis so that we can assign workflows and get those things going. And I think the workflows look really promising for that. It's been a struggle for us to get people up to speed at the pace and scale at which we do things. And so getting people all on the same page and having that is going to be really awesome as we go forward, building templates and being able to smooth that. And then the ELN too, I told a story to Hannah just the other day where, we basically learned that we had the capacity to do this. And I mentioned it at lab meeting. And the next time I came into our lab room, we had two students really playing with it. And when I wasn't back in time to help them with their issue, they sent me a lab notebook to review. And I got an email that I was supposed to review an ELN from LabKey. And I was like, that's weird. And I opened it and it just said, Dillon, come back. We need your help. And so they were spending time playing with the ELN after they figured out that we could use it, because they're very interested in this, right? The buy-in's there. They know that it's going to make things easier in the long term, and they want to learn how to use it too. Especially things like PCR amplification plots, right? Not having to print and tape figures into paper lab notebooks, being able to associate it with data items and objects that we have already entered is just going to be really huge, especially with so many different types of samples and assays. I think I'm probably went a little bit long, but I appreciate the opportunity to come talk to you all, and we can see if there's questions.
Awesome. Thanks so much, Dillon. That was a really great presentation. I love that the lab is using ELN maybe as a chat feature as well. That's something for us to consider in the future.
I know. I was like, that's not what that's for. They were like, yeah, but we figured out how to do it. And
Yeah.
like, yes, I'm happy as well.
Yeah, we'll take it. So I've got a question, and then there's a few questions in the chat here. So you mentioned how useful it would be to have a cloud-based system where you could kind of do data entry more remotely, kind of outside of the confines of the lab itself. So can you talk to me about how that process has been for you with entering data in the field?
We have not transitioned to field data entry yet. I know in one of our earlier meetings, Andy mentioned the prospect of like an app, right? Once upon a time in like 2010, we experimented with taking iPads out in the field to help with duck species identification and data entry. And the tech and integration just was not there yet at that time. And I think it's something that we haven't lost sight of. And, I think you gave us oh yeah, like we've heard a mobile app could be useful and getting there in the future. And, I think especially as we turn towards maybe working on the ELN and working on getting laptops next to our biosafety hoods that we can work in real time with that. Once we get to the mobile device platform to take into the field, it'll be, I think, easy to get on there. We're not quite there yet, but I can see it happening.
Awesome. Yeah. And of course I ask because we are interested in doing that in the future and looking to identify, common pain points there.
Yeah, exactly.
Okay. So a couple other questions here. It sounds like the work that you're doing requires lots of collaboration and coordination. How do you use your tools with others outside of that, outside of your organization?
Ooh, such a good question. It does require a lot of collaboration, coordination, especially, in a way in the scientific process, we produce a ton of reagents and biospecimens, and part of our role as in contribution to the public knowledge bank is sharing those with collaborators and finding other research questions to address with them. So that's been something we've done for a long time, and data sharing has become more complicated, right? I'm sure everyone here has done the institutional data trading because I know it's mandatory at Ohio State. which makes it weird and difficult to share those things. But what I love about LabKey is especially, oh man, the new feature of being able to save views, right, and rearrange your columns was really just like I was a kid in a candy shop, because being able to save different views for different types of data exports and different reporting, when in reality, a lot of it is exported as a CSV, blind this, and then get it to a collaborator so they can now analyze it. Being able to save like how exactly I'm blinding that and which data fields I need and what order this person prefers them in, for example, I think it is really cool. I think at some point in the future, getting to a point where our longstanding collaborators and getting them access to the database itself so that they can work with us in more real time will be a cool next step as well.
Yeah, awesome. That's great. And then maybe we've got time for one more question here. The first one is a statement. You seem really organized. How much time do you think your group spends on data management right now?
I'm
It's a loaded
glad you
question.
think I seem organized. I certainly don't feel organized most of the time. It's interesting too, especially the growing pain side of this, right? Is that it felt like there were 6 months I was the only person doing data management, and I'm a PhD candidate, right? I'm not a data manager. I'm not a full-time research staff. And I just happened to it out of the people who we had in there at that time. And so a lot of the burden is sort of trickled down, and it's need based. It's hard to say exactly how many hours we probably spend across the lab group now. I would say collectively between all of our person time, data entry, we're probably alone spending about 20 hours of person time a week, just as that MINC project is samples that come in on a weekly basis, right? So receiving samples, getting those data sheets out, getting them entered into the database in real time is a challenge. As far as management on the back end, probably more like 5 to 10 person hours per week. And it depends on who's working on which manuscript and what the analysis priorities are at that time.
Totally. Well, great. Thank you so much again, Dillon, for presenting. That was really fun to see.
Thanks for having me.